So in this lecture, we are going to talk about adrenal gland physiology. Then we'll have a separate lecture talking about adrenal gland pathophysiology. So first of all, it's important that we review the anatomy of the adrenal gland. So we see that the adrenal gland is made up of two important parts. We have the cortex and the medulla. The medulla is usually chiefly composed of chromaffin cells which produce catecholamines, i.e. epinephrines and norepinephrines, while the cortex is usually made up of three layers: the zona fasciculata, the zona reticularis, and the zona glomerulosa, which are important for production of different cortical hormones. So in the microscopic section below, we can see that we have the medulla at the far lower end. Then you have the zona reticularis, fasciculata, and glomerulosa. So, sorry. For the zones of the adrenal gland, number one, it is divided into the adrenal medulla and the cortex. The medulla is usually composed of chromaffin cells, while the cortex is composed of, number one, the zona glomerulosa, which forms the outermost layer, the zona fasciculata, which forms the middle layer, which is usually wide, and finally the zona reticularis, which forms the innermost layer. So this one is just a repeat of whatever we have talked about. So number one, we start with zona glomerulosa. It is the outermost cortical zone of the adrenal cortex. It usually makes up approximately 15% of the entire cortex, of the cortical volume. Then it chiefly secretes aldosterone hormone. This is very important. And for us to produce aldosterone hormone, then we need to have stimulus. And the stimulus for production of aldosterone is usually angiotensin 2 and potassium ions. So this aldosterone, we are going to see that it is usually made from cholesterol, and it requires an enzyme called aldosterone synthase. So aldosterone synthase enzyme is usually found in the zona glomerulosa of the cortex. So secretion of the zona glomerulosa is usually influenced by angiotensin 2 and potassium ions. But again, we'll see later on that even ACTH in a very little way, can influence the production of hormones from the zona glomerulosa. But you need to remember that the most important ones are angiotensin 2 and potassium ions. The zona fasciculata is the middle and the largest layer of the adrenal cortex, and it makes approximately 75% of the cortical volume. So you can imagine how large it is. So it usually secretes chiefly glucocorticoids, which include cortisol and corticosterone. But again, you can have adrenal androgens and even estrogens, which come into play. So the secretion of the zona fasciculata is usually under the influence of ACTH. And ACTH is usually produced from the anterior pituitary gland under the influence of CRF, corticotropin releasing factor from the hypothalamus. The zona reticularis is the innermost layer of the cortex, and it is important in the production of adrenal androgens. And we have dihydroepiandrosterone and androstenedione. But again, it can secrete small amounts of estrogens and some glucocorticoids, but importantly, it secretes the two adrenal androgens. So the production of hormones in the zona reticularis is usually under the influence of ACTH, but again, cortical androgen stimulating hormones can also come into play to help in stimulating this particular region to produce the different hormones. So then it will be important for us to talk about the process of steroidogenesis. And this is basically production of hormones from cholesterol. So this cholesterol here can be gotten from the diet, but again, it can be made from the body de novo. Now, the first step, which is usually the rate limiting step of steroidogenesis, is the conversion of cholesterol to pregnenolone. And here we require an enzyme called cholesterol desmolase enzyme. Now, after forming pregnenolone, we can have two pathways. One pathway going down to form aldosterone, and the other pathway can go to form cortisol and the adrenal androgens. But again, you can see that even in this pathway for formation of aldosterone, one of the intermediates, progesterone, can be taken into the other pathways to help in formation of cortisol and androgens. So it is really important that a student remembers the enzymes that are important in these pathways, because when we'll be talking about the pathophysiology of this process, we're going to talk about a condition called congenital adrenal hyperplasia. And it will be very important for us to understand where these enzymes are, these hormones are, what they do, and what will happen in case we block them. For example, the most common enzyme affected in congenital adrenal hyperplasia is usually 11 beta hydroxylase enzyme. So the moment we block, or rather when we mutate the gene that codes for this 11 beta hydroxylase enzyme, then it means that we can't form aldosterone and we can't form cortisol. So this entire pathway will overflow to produce the androgens. Now, congenital adrenal hyperplasia is usually associated with virilization of females. But in male, there's usually no significant change because they have the most important testosterone hormone definitely from the testes. But we will talk about that when the right time comes. So we start with the mineralocorticoids. Number one, we have aldosterone, which is the most potent and accounts for approximately 90% of all the mineralocorticoid activity. Then you have deoxycorticosterone, which usually accounts to 1/30. No, sorry, deoxycorticosterone is 1/30 as potent as aldosterone, but it is usually released in very small quantities. Then again, corticosterone, 9-alpha-fluorocortisol, which is a synthetic, and is usually more potent than aldosterone. And again, you have cortisol and cortisone. All of them have mineralocorticoid activity, but it depends which one has a stronger one, potent one, and which one has a weak or a slight activity. So aldosterone is usually produced from the zona glomerulosa, as we had seen before, and the stimulus for production of aldosterone is usually angiotensin 2 and potassium ions. So in the pathway that we have to the right, we can see that we have cholesterol being converted to pregnenolone, of course, the rate limiting step. Now, after forming pregnenolone, we can form progesterone using the enzyme 3-beta-hydroxysteroid dehydrogenase, which now can go further down up to a point where we produce aldosterone. This enzyme is supposed to be aldosterone synthase, not androgen synthase. Sorry for that. Now, for the cellular mechanism of aldosterone, number one, this hormone is lipophilic. So it can cross the cell membrane and enter the cell to the cytoplasm. Now, for that issue, they usually have cytoplasmic mineralocorticoid receptors. They usually diffuse, so they pass through the cell membrane to the cytoplasm where they combine with their receptor to form a receptor complex. Now, after forming that aldosterone mineralocorticoid receptor complex, this usually diffuses to the nucleus of the cell. Now, once it has gone to the nucleus of the cell, it can influence a lot of things happening around the nucleus. Number one, it can cause transcription and formation of messenger RNA, which can later be translated from different proteins. For example, a good one being sodium potassium ATPase, which is very important for transport of sodium and potassium. Again, I think I've talked about that point. So another thing to understand is that the mode of action of aldosterone is usually quite slow because it involves formation of a new protein. Compared to other hormones which act via a cell membrane receptor, any hormone that acts via interference with the gene transcription will take a lot of time because it needs to form a lot of proteins in form of enzymes, which can even take like around 1 hour before they can help in bringing a change in the body. So this diagram here is trying to show us the mechanism of action of aldosterone. Here we have the tubular lumen, and here we have the renal interstitial fluid. You can see aldosterone is passing into the cell. Of course, it is lipophilic, after which it combines with the mineralocorticoid receptor to form an aldosterone mineralocorticoid receptor complex. Now, this complex can influence gene transcription, forming a myriad of proteins, one of them being a sodium ion channel. But again, we can form the sodium-potassium ATPase, which can help in regulation of the levels of potassium and sodium ions in the ECF. Sorry. Now, how do you regulate aldosterone secretion? Number one, we always have increased potassium. And number two, we have angiotensin concentration. So when these ones are increased, we are going to have production of aldosterone. But when the levels of sodium ion increases in the ECF, then we are going to decrease aldosterone secretion. Why? Because aldosterone secretion is very important for the reabsorption of sodium ion. So because already we have high sodium ions in the ECF, there's no need of adding to whatever we have. So we need to slow down the production of this hormone. In cases of hypertension or hypervolemia, we usually have the atrial stretch. The atrium of the heart usually stretches. And when the atrium stretches, they usually release a peptide called the atrial natriuretic peptide ANP. And this hormone can also help in reduction of aldosterone secretion. Then again, we should know that ACTH from the anterior pituitary gland can also influence aldosterone secretion, but this one is usually contributing to only a little effect, as I had alluded to before. So the functions of the aldosterone still, we have it increases renal tubular reabsorption of sodium and secretion of potassium. You remember whenever we have a sodium potassium ATPase, which definitely is a co-transporter, it will be reabsorbing sodium ion back into blood, while at the same time, it will be pumping potassium ions into the tubular lumen. And that's why we say that it will increase reabsorption of sodium ion, while it will increase secretion of potassium ion through urine. Now, this action usually occurs at the principal cells of the collecting tube. So at this point, we can see that excess aldosterone will lead to increased ECF volume, following the fact that a lot of sodium ions will be reabsorbed. This will increase the osmolarity of the ECF, hence water will flow into the ECF by osmosis. But again, intriguingly, is that aldosterone deficiency can cause hyponatremia, where we have low sodium levels below 135. Excess aldosterone, again, is associated with pressure diuresis and pressure natriuresis, what we otherwise call aldosterone. Now, if I can talk about this, you see, when we reabsorb sodium ions back into blood or back into the ECF, and we reabsorb water, we're going to expand the blood volume. Now, someone might expect that in case we have excessive aldosterone, as occurs in some conditions we're going to discuss later, then water will be continuously reabsorbed into our body. But this one doesn't always occur. A point reaches when your body has a lot of water due to excess aldosterone. Now, because of that, the kidney will come into play and start secreting a lot of urine. Now, in the act of secreting a lot of urine, what we otherwise call pressure diuresis, you can also have pressure natriuresis, where we also excrete a lot of sodium ions out. Now, this is usually called aldosterone escape, where the body tries to escape the wrath of excess aldosterone. In patients with zero aldosterone, they usually suffer from circulatory shock, which can lead to death, because remember, aldosterone is very important for maintaining the amount of blood in the system. So in case you don't have it, you're going to have hypovolemia, which can later on lead to circulatory shock and death. Now, at this point, you can see that excess aldosterone can cause hypokalemia and muscle weakness. And we know that calcium, potassium ions, and sodium ions are very important for action potential generation. So when you have low levels of potassium ions, you're going to have muscle weakness, because even the action potential generation won't be that efficient. And that's why in patients suffering from esophageal cancer, some of them do vomit a lot because of obstruction of the esophageal canal. And when they try to swallow saliva, they feel a lot of pain. So some of the patients usually prefer to maybe remove that saliva via their mouth. Now, when they do so, they usually also lose a lot of potassium ions, because from our GIT physiology lectures, we see that One of the most important components of saliva is potassium ion. So the moment you're vomiting a lot, then you can suffer from hypokalemia. But again, aldosterone deficiency can cause hyperkalemia. Now this can lead to cardiac toxicity because remember, potassium ion is one of the most important cations in the process of action potential generation within the cardiac tissue. Again, excess aldosterone can lead to alkalosis. This one is usually due to interference with the intercalated cells of the collecting tubule. So whenever you secrete a lot of hydrogen ion, you're going to have alkalosis, usually associated with high body pH. So we go to cortisol as a mineralocorticoid. This is quite important. So remember, cortisol has a weak mineralocorticoid activity from our introduction. Actually, it is 3,000 times less than aldosterone in terms of its potency as a mineralocorticoid. But now something interesting is that it is 2,000 times more abundant than aldosterone. So quantitatively, you can see that cortisol has a relatively potent mineralocorticoid activity owing to the fact that it's really abundant in the body. So again, it can bind to the mineralocorticoid receptor with a very high affinity. So we can't really assume that it can't have significance in the body. It does as a mineralocorticoid. But the body doesn't allow that to happen because already you have aldosterone doing the work. So we usually have an enzyme called 11-beta hydroxysteroid dehydrogenase type 2. And this enzyme is usually very important in modulating the levels of cortisol. So we're going to see how that usually happens in a few. So cortisol can be converted to cortisone, and cortisone is usually inactive at the pre-receptor level in the renal tubule. Hence, this will protect the mineralocorticoid activity receptor from activation by cortisol. Now, we have said that we have isoforms of 11-beta hydroxysteroid dehydrogenase. Now, we have an enzyme called 11-beta hydroxysteroid dehydrogenase type 2. This enzyme usually converts cortisol to cortisone, and cortisone is usually inactive at the receptor level, so it can't activate a mineralocorticoid receptor. Now, this one is usually so because we want to try in as much as we can to prevent cortisol acting as a mineralocorticoid. But see, in some patients, we always have deficiency of this enzyme. So because we have deficiency of this enzyme, it means that patients can actually have cortisol in their body acting like a mineralocorticoid. Now, when this happens, we call it apparent mineralocorticoid excess syndrome, AME. So what are the causes of this apparent mineralocorticoid excess? Number one, we have genetic mutation in the gene coding for 11-beta hydroxysteroid dehydrogenase type 2. People who ingest a lot of licorice can also be suffering from this AME. But again, in Cushing syndrome, because we're producing excessive amounts of cortisol, this can actually activate the mineralocorticoid receptor due to their abundance. Imagine a normal physiological apparatus, we have 2,000 times more cortisol than aldosterone. So imagine when you have Cushing syndrome, what can be the ratio? So this is trying to show us what is going to happen, how the body prevents this cortisol from being a mineralocorticoid. Now at the kidney, colon, sweat, and salivary gland level, we always have cortisol, which is inactive form, being converted to cortisone, which is inactive. So the enzyme being used is called 11-beta hydroxysteroid dehydrogenase type 2. Now this cortisone, though it has a weak mineralocorticoid activity, but it can't bind to the mineralocorticoid receptor. So by converting cortisol to cortisone, you're going to prevent it from acting like a mineralocorticoid. Sorry. So this cortisone can go to the liver, skin, brain, adipose tissue, and the placenta, where they get the first isoform of 11-beta hydroxysteroid dehydrogenase, which is type 1. So this type 1 will convert cortisol, which is inactive, to active cortisol. Now this cortisol can bind into the glucocorticoid receptor and enact its glucocorticoid activity. Now when using this knowledge, you can see how we can regulate cortisol from acting like a mineralocorticoid hormone. But again, we can see that we can still maintain its potency of being a glucocorticoid hormone. So for glucocorticoids, we have cortisol, which is very potent, approximately 95% of the glucocorticoid activity. We have corticosterone, which provides approximately 4% of the total corticoid activity. Then you have the synthetic forms like prednisone, which is 4 times as potent as cortisol, methylprednisolone, dexamethasone, as you can see, which is 30 times as potent as cortisol. And actually, I'm forgetting to say this, that in so many research that we use dexamethasone has the potency of lowering the immunity of the body to very low extent. Hence, the animal model being used can die of immunosuppression because of some opportunistic infections of some sorts. So cortisol is a stress hormone usually produced from the zona fasciculata under the influence of ACTH. And it's also called hydrocortisone, so they are synonymous. So this pathway basically tries to explain what is going to happen for us to form cortisol. So we had talked about this pathway, but you can see that these products can be channeled into the second pathway. In an exam, you need to know that pregnenolone is the common product in all the pathways because it serves or it contributes to all the pathways. But this pathway is where we are forming our cortisol. And you see we have two important enzymes, 21 hydroxylase and 11 beta hydroxylase enzyme. They are very important for formation of cortisol. For the cellular mechanism, the same thing as that one we saw in aldosterone. Cortisol is a lipophilic hormone, so it can cross the cell membrane to the cytoplasm. Now, once in the cytoplasm, it can bind to its cytoplasmic receptor to form a hormone receptor complex, which then can move to the nucleus and interact with the DNA sequences. Now, when we have that interaction, we can either activate transcription or we can repress transcription. So in doing so, we can regulate the number of proteins within the cell, and we can have a myriad of effects within the body. So this basically tries to summarize what I'm talking about. We can see we have a glucocorticoid, i.e. cortisol, diffusing in, combining the receptor. We have chaperone basically for folding of the protein and unfolding. Then after that, we can now take the hormone receptor complex to the nucleus where it can either activate or repress gene transcription. So we talk about regulation of cortisol secretion. So the stimulus for production of cortisol is usually stress. And this stress can come from things like surgery, infections, trauma, maybe necrotizing substance injected under the skin, etc., even a bite. So whenever we have stress, we're going to excite our hypothalamus, which is going to produce CRF, corticotropin releasing factor. This is going to be carried to the anterior pituitary gland, which will influence it to produce ACTH, otherwise called corticotropin. Now, corticotropin will now be carried to the adrenal cortex, especially the zona fasciculata, and stimulate it to produce cortisol. Now, this cortisol can have a myriad of activities like gluconeogenesis, protein mobilization, fat mobilization, etc. But interestingly, for us to regulate the amount of cortisol, we usually have something called the negative feedback loop. So we have the short negative feedback loop to the anterior pituitary gland and the long negative feedback loop to the hypothalamus. So whenever the amount of cortisol in the body is excess, we usually exert a negative feedback loop to the anterior pituitary gland and to the hypothalamus. Hence, in high levels of cortisol, we expect to have a low levels of CRF and low levels of ACTH. And that's why in tumors or adenomas of the adrenal gland producing high amounts of cortisol leading to Cushing syndrome, we actually have low levels of ACTH and CRF because of the negative feedback loop. So the chemistry of ACTH and CRF, not that important, but CRF is just composed of 41 amino acids, while ACTH is composed of 39 amino acids. So these are the factors that stimulate cortisol release. Notice all of them will lead to stress. Even cold can cause stress within the body, things like surgery, etc. Even straining an animal from moving, especially when you strain a rat from moving, imagine. It will be under stress. It will be trying to move. Now that can be a stimulus for production of cortisol. And again, even in some debilitating types of diseases, we can also have cortisol being released. So the effects of cortisol, number one, is to stimulate gluconeogenesis. And basically, this will involve anything revolving around gluconeogenesis. We can have increasing enzymes for converting glucogenic amino acids to glucose. We can mobilize amino acids from the extrahepatic tissues. But again, importantly, cortisol usually antagonizes insulin's effect. This is very, very important. It usually antagonizes insulin's effect, so it is anti-insulinic. Others are glucagon, and even epinephrine can also be anti-insulinic. Now, one intriguing thing about cortisol is that it will increase the blood glucose level, but it will decrease its utilization. And one of the mechanisms that has been perpetuated is that it can reduce expression of GLUT4 in the muscular tissue. So glucose cannot be used by the muscle, and thus it will accumulate within your blood. Again, it can elevate blood glucose level concentration, and it can cause adrenal diabetes. Adrenal diabetes is diabetes is basically high glucose level. So when you have cortisol, you can see that you increase the level of glucose in blood, but you reduce its utilization. So in a way, you're going to cause diabetes, and that's why you call it adrenal diabetes, because it is purely due to a secretion from the adrenal gland, i.e. cortisol. It can decrease protein synthesis, but increase protein breakdown, as you have seen, to mobilize amino acids. And again, cortisol increases liver and plasma proteins. Another important thing is that it will increase blood amino acids, but it will diminish transport of these amino acids to extrahepatic tissue, but it will enhance transport of these amino acids to the hepatic tissue. So in short, cortisol will give the hepatic cells a more priority than the extrahepatic cells. Again, it can mobilize fatty acids, and usually it causes obesity. Now, this is usually paradoxical from what we know. Imagine cortisol can break down your protein, it can mobilize fatty acid, it can break down glucose, but outcomes it can lead to obesity. Now, when you read different types of books and even papers, they say that even though cortisol can lead to all these things that I've talked about, but it usually stimulates food intake. And that's why a person will have obesity. Yes, I'm breaking down a lot of things in your body, but you're eating a lot. Now, how do you describe this obesity? You say that for cortisol, the obesity is a buffalo-like torso and a moon-like face. That's how we describe obesity due to maybe Cushing syndrome or excessive amounts of cortisol. Now, it has anti-inflammatory effects. This is very important. So in any time you're doing any question about a cortisol hormone, it will reduce the levels of inflammatory cytokines, things like interleukin-1. Again, it will downregulate the number of T lymphocytes, B lymphocytes. It will lower the number of neutrophils, everything, because it will be lowering inflammation, sorry. Again, cortisol can help in resolution of inflammation. So in case you had inflammation, cortisol can help to end that inflammation from continuing. Again, it can block inflammatory response to allergic reactions. I think I've talked about this one. It can reduce the number of eosinophils lymphocytes. And again, it can cause atrophy of the lymph nodes, because a lymph node is very important as a secondary lymph node organ. Cortisol can reduce the levels of interleukin-1, which we see plays a very important role in terms of fever. So you can really relate that and see how it happens. Now we go to this hormone called corticotropin or adrenocorticotropin or ACTH. Now, this hormone is very, very interesting. Number one, it is usually released from the anterior pituitary gland under the influence of CRF, corticotropin releasing factor. Now, ACTH usually activates adrenocortical cells to produce steroids by increasing cyclic AMP. So we've seen that cortisol and aldosterone act via the nucleus, but ACTH will act at the cell membrane level. And the only way it is going to work is by increasing the levels of cyclic AMP. Now, we have said that these hormones usually increase during stress. So in case you have any physiological stress of any sort, you're going to increase ACTH secretion. Now, basically, this is what is happening, ACTH acting to the receptor, increasing the levels of cyclic AMP, which has a myriad of effects, but eventually activating this enzyme called cholesterol desmolase enzyme, which is very important for producing pregnenolone. Now, from there, I hope you know the pathway, how it continues. So we go to something interesting and rather controversial. Now, whenever we are producing ACTH at the anterior pituitary gland, we usually produce it together with other small hormones like melanocyte-stimulating hormone, lipotropin, and endorphin. So ACTH is usually secreted by the anterior pituitary gland with several other hormones that have a similar chemical structure. And the gene that is transcribed to form ACTH usually produces a more larger protein called a pre-prohormone. And this pre-prohormone is usually called the pro-opiomelanocortin, POMC. So you can see that this is the gene that is, this is a gene coded for the ACTH production, but first of all, it formed this protein. Now you see after this particular point, we have different sections. One of them can produce the gamma MSH, others can produce lipotropin, others can produce endorphin, lipotropin, beta MSH. But you see this section is producing ACTH. I want us to notice the following. ACTH is composed of two parts. It has the alpha MSH part and the CLIP part. So this is called melanocyte stimulating hormone. So ACTH, because it has a sequence for MSH, it can also have some melanocyte activity. It can help in melanin production. The question then becomes between MSH and ACTH, which one will help in production of melanin? Because we also have the gamma MSH, as you can see, but this ACTH again has some alpha MSH. So which is which? Now for pigmentation, it is worth noting that even though MSH is more potent, or the gamma MSH is more potent, or the pure form of MSH is more potent in terms of helping to produce melanin, but it is usually quantitatively less within your body. The levels of pure MSH in your body is extremely small. And thus, it is thought that pure MSH doesn't contribute to melanin production in the skin. But because ACTH, as we have seen before here, it has an alpha MSH activity. And again, ACTH is 30 times. I think it is very, very abundant compared to MSH, pure MSH. It is believed that ACTH usually helps in production of melanin. Number one, because it has a very weak melanocyte stimulating effect because of the MSH sequence that you have seen. But even importantly, because it is quantitatively more than a pure MSH. So it is thought that it can help in that role. So that is the end of our lecture today.